everybody. My name is Carlos Souza. I'm the CEO of Immunicum. We are a publicly listed company in the main market, Nasdaq Stockholm. So I need to show you the slide. And what is Immunicum? Immunicum is a biotech company based in Sweden and working in the immuno-oncology space. We have quite a unique immune primer that is well advanced in development. It's a cell-based therapy, but is an off-the-shelf and allogeneic cell-based therapy. For the ones that are familiar with the space, this is a very big differentiation. We are quite well advanced in phase 2. We are going to have results of our first phase 2 study third quarter this year, so very, very close. We have, now in different indications, more than 90 patients treated with ilixadencel and showing a very good safety profile. Basically, the side effect that we have is fever treated with paracetamol. As you will see, for the mechanism of action, we induce an inflammatory response, so this is expected. We also have quite a robust manufacturing process for development. But we are initiating now the development activities to develop a commercial process. We have had several interactions with both the European authorities and the FDA. We have this phase 2 study I mentioned to you is in Europe and US, and we have now a new study that has just initiated primarily in the US. So we already went to the FDA twice with 2 protocols. So very good feedback from both set of authorities. Recently in November, we also announced a development supply agreement collaboration with Pfizer and Merck Serono, where they will supply us at no cost the checkpoint inhibitor for the phase 2 of our new study. We just concluded in December the raise of $39 million. This was done with a mix of directed issue and rights issue. And also through this financing, we brought into the company a very strong group of institutional investors that have a much longer time frame in terms of investment, very important for the company. And this financing, together with the cash that we had in the company, will fund us towards the end of 2021. And this runway will take us across very big potential value inflection points. The team is very experienced in all the different sets of the space of immuno-oncology. So briefly, our pipeline, we have 3 projects, ilixadencel in the clinic, where I will spend more time, and 2 additional projects in preclinical. As you can see, the most advanced study is in kidney cancer. We also have a study in liver cancer, in gastrointestinal stromal tumor, and now we are starting the new one in head and neck, lung, and gastric adenocarcinoma. But, where do we fit in the landscape, very busy landscape of treating cancer, in particular when you look at the immuno-oncology space? On your right, you have all the classes, checkpoint inhibitors, kinase inhibitors, even some chemotherapies, classes that block the defenses that the tumor cells have against the immune system. As you know, the big challenge with these classes have been the fact that if you don't have a hot tumor, if you don't have an active presence of a significant number of immune cells and they are activated, you don't get the expected efficacy. So we operate on the left side. We prime the patient's own immune system, so you can get the synergistic effect when you combine. So we can be combined with kinase inhibitors, checkpoint inhibitors, even some chemotherapy. Key differentiation, as I mentioned, to other cell-based therapies is the fact that our cells are originated from healthy donors. So these are normal blood donors. It's just instead of taking full blood, we do leukapheresis, extract monocytes, transform into immature dendritic cells, and then we activate them with a cocktail of cytokines and make them extremely pro-inflammatory. When they start producing chemokines and cytokines, we cryopreserve them. From one donor, we treat about 50 patients. So as you can see, very different from other cell-based therapies. and also the fact this fact has an impact on the cost of goods that has no impact on the margins. The batches are frozen and basically can sit in the hospital pharmacy. We already have shelf life passing the 3 years. So, we can produce whenever we want, and we store and readily available to be administered to the patient when needed. We currently produce in Germany, and now with part of the process, as I mentioned, we are tech transferred to a different CMO with commercial capabilities. So the product, 1 ml vial, about 10 million cells, sits in the hospital pharmacy. When needed, you just thaw it to bring it to, of course, the body temperature, and then you inject the cells intratumorally. You can inject the primary tumor, the metastasis, it doesn't matter, but you just need to inject one of the masses. You don't need to do multiple injections. The activity is not going to be a local effect. The mechanism of action is to prime and kickstart the inflammatory process. So when you inject these donor dendritic cells that were activated into the tumor, they start releasing chemokines and cytokines. TNF-alpha, IL-1 beta, and this is going to induce a very strong recruitment of patient-own immune cells. The first ones are the NK cells, massive recruitment and activation. They kill tumor cells, and here you do release of neoantigens. So elixir cell by itself is what we call a universal product. You don't need to do any matching. Use the same cells regardless of the type of tumor, regardless of the patient. But when you inject it and you initiate this cascade, you are going to make that reaction personalized. So these neoantigens that are released are taken over by the patient standby dendritic cells that come also to the site of the injection, take on the neoantigens, are matured faster through interferon gamma, migrate to the lymph nodes, and here they do the education of the tumor-specific CD8 plus T cells. The donor cells are destroyed in the first 2 to 3 days. So they did their job, release chemokines, cytokines. They are also foreign cells, so they amplify the immunogenic reaction, but then all the activity is done by the patient's own immune cells, and that's spread through the whole body. Here also one of the reasons for the excellent safety profile, because again, as I mentioned, you don't have donor cells in circulation and causing some cytokine reactions. First study already in 2012, before all the checkpoint inhibitors were even a talk. Newly diagnosed metastatic renal cell carcinoma patients. What you see here is these patients were elected to surgery to remove the kidney. Before that, we were able to inject with 2 doses. So day 1, day 14. So patients with renal cell were treated with just 2 doses, and the administration is like if you do a biopsy, so no requirements for any specific techniques or equipment. What you see here is on your left side as a reference, what you see as a normal reaction. The dot spots are the CD8 plus T cells. The other images are from one of the patients in the study, so you see the massive infiltration of CD8 plus T cells that are intratumorally. These are cells that were educated in the lymph node and then came to the primary tumor. Very specific, you see the healthy tissue just outside tumor as the normal concentration of cells, but also very important is that you also have the subscopal effect. That is a metastasis, as a matter of fact, was a lung metastasis that also get a very strong infiltration of these tumor-specific CD8 plus T cells. From a perspective of the clinic, the study had 11 patients. We could show that, we published this data, we have median overall survival of 4 years. So it's not a big study, but quite an extended survival. The patients treated with sunitinib are expected to reach 14 to 16 months of survival. On top of that, we have, in last time we could check, because of GDPR, a year ago, 3 patients still alive and passing the 5-year survival mark. So again, not big numbers, but quite impressive impact on the patient survival, particularly considering that half of these patients had 9 months expected survival because they were poor prognosis patients. One of those patients is this one in this case. We use this really to show the benefit of having classes of drugs with a different mechanism of action and a synergistic effect. This patient had, again, a poor prognosis. He had several lung metastases, several liver metastases, and then he developed 4 brain metastases. Remember, we're talking about 2012. The only alternative was sunitinib, that is very well known, that doesn't treat brain metastases, very well documented in the literature. Anyway, this patient then resolved the brain metastasis disappear, liver metastasis disappear. He had a small lung metastasis that was removed surgically, the one you saw, the histology. For this last capital raise, we were organizing meetings to our shareholders, and then this gentleman, this was in October, stand up and say, I'm this patient. So he traveled for 3 hours to come to our meeting, and this is why we know, we have a more updated information, that in October he was still alive after 66 months, and he is having a normal life. Again, we are not trying to make it more than what it is one case, but here you don't see brain metastasis disappear spontaneously, and here is really showing the benefit of combining classes of drugs with a complementary and synergistic mode of action. This took us to then run a phase 2 study, a MARECA study, but this is a comparative study. We recruited 88 patients in 8 European countries and the US. 58 of them is a combination of ilixadencel plus Sutent, and 30 of them is Sutent alone. So this really gives us 2 comparative arms. It's a survival study, so 18 months follow-up. Last patient first visit was January last year. This is why we are expecting now to have the top-line results of the study in third quarter this year. This is a very important study. It's the first one where we have comparative arms. And again, we are talking with usual suspects as potential partners, and this is the type of data that they are waiting for. We also just recently published the results of a liver cancer study, hepatic cell carcinoma, where we show also the immune activation, the same type of safety, and some initial indications of efficacy, even in second line as mono. So we would like to further proceed, develop an indication. But the big study where we are focusing now, of course, MARECA, the phase 2, very important for the company, but running. But now we, of course, naturally, we wanted to combine with checkpoint inhibitors. So we selected head and neck, non-small cell lung cancer, gastric adenocarcinoma. Very big indications, very poorly served by the checkpoint inhibitors. So phase 1B, so we already activated US centers in combination with Keytruda, 21 patients. This is going to give us already this year safety data. Very important, because if we maintain the safety profile, we are the only class of drugs, the only product that combined with the checkpoint inhibitors, you don't have added toxicity. So it's already going to be very important, but also some potential initial indications of efficacy because it's 21 patients. When you move to the phase 2, these 3 indications run in parallel as 3 different studies. And this is where in non-small cell lung cancer, we continue with Keytruda, the collaboration with Pfizer Merck Serono, in addition to give us scientific validation, because both of these companies have very few of these collaborations, and they accept to be part of this development collaboration. Also allows us to expand the study into Europe, and this, of course, operational will make it easy for us. We also recently at ESMO had a poster presentation showing the synergistic effect in preclinical studies in combination with checkpoint inhibitors, but also with a class of the immune enhancers. Very exciting results, and we're going to continue exploring those avenues. As I mentioned to you, recently concluded financing, $39 million, very big institutional investors in Sweden and Scandinavia, providing us with a very stable financial situation, allowing us to pass these different value inflection points. So in 2019, very, very big year for us. We are going to get the top-line results of the small orphan disease gastrointestinal stromal tumors. we're going to get the phase 2 miracle results. This can be transformational for the company, but also the initial data on the new study in combination with checkpoint inhibitors. 2020, results of the phase 1b can already give us some of the initial indications of efficacy. And again, depending on the speed of enrollment, potentially some initial results from the phase 2. So, in a very short period of time, we are going to pass very strong data that, of course, we are expecting to be positive, and this can be transformational. Because, as you know, Big Pharma has been primarily betting on the blocking the defenses, checkpoint inhibitors. They are lacking on primers, and you know that last year was a very active year for acquisitions of companies with immune primers. So we are part of that. We have a unique mechanism of action. No one else is doing it. We have a patent situation, strong patent situation. On top of that, we are a ATMP, what gives us additional protection. So, in summary, as an investment case, we have all the pieces in place. We have the financing to take us to a very healthy runway across very significant value inflection points. We have an experienced team in place. We have now a good mix of retail investors, but strong institutional investors. And part of my activities is now really to expand even more the geographic reach of these investors. We have a clearly defined strategy. Despite the fact that we already have quite a robust development process, we are also initiating the development of a commercial process. And this will take us, of course, save us a very valuable time for the initiation of pivotal studies and for a regulatory approval. Because we have been having in several interactions with the health authorities, both the European and the FDA, we have a very clear understanding, and they know us in terms of what is the avenues moving forward. So we are, thank you for your attention, and we are looking forward for a very exciting 2019 for the company and for this space too. Thank you.